The last phase of mitosis is telophase. During telophase, the chromosomes are at one end of each end of the cell, and the nuclear membrane begins to reform around those chromosomes. The chromosomes unwind, and when they're all unwound and the nuclear membrane is reformed, mitosis is complete. So let's take a minute to draw our drawing for telophase. You should have some really short little spindle fibers coming out on both sides of the cell. So since we're working with 4 chromosomes, I'm going to draw 4 short little spindle fibers coming out. And then at the end of each little spindle fiber, we should have our chromosome, or our sister chromatid. So the most important part about telophase is that the chromosomes have all been brought to the other side of the cell, and then we have the beginnings of the new nuclear membrane forming around these chromosomes. Now, the nuclear membrane isn't completely formed, but it's starting to reform. And that's it for telophase. Let's take a look at those Cornell notes. How are they coming along? Let's pause if you need to. The last stage in the cell cycle is cytokinesis. Cytokinesis is the process in which the cytoplasm and the cell cleave or divide in two. The parent cell becomes 2 new daughter cells, and the cell cycle begins for the 2 new daughter cells. So let's take a look at what cytokinesis looks like in our drawings. You can see that it results in 2 separate daughter cells. Cytokinesis in animal cells is a little bit different in plant cells. In animal cells, the cell membrane pinches inward and forms a cleavage furrow, but in plant cells and in other cells like protists, a new cell wall has to be formed. So a cell plate forms in the middle of the cell and grows to the ends to make 2 new cells. Here's a picture of some plant cells in various stages of mitosis. Can you guess which stage each one is? This first stage, where the chromatin is loose and unwound, is interphase. We go to the next stage, where the chromosomes are condensing into rod-like shapes and the nuclear membrane is beginning to dissolve. That's prophase. Then we can see the chromosomes lined up along the equator of the cell, so that's got to be metaphase. In this next stage, you can see the spindle fibers pulling the chromatids apart. These ones going to one side of the cell, and these going to the other side of the cell. So that must be anaphase. And then lastly, we can see the chromosomes unwinding, the nuclear membrane reforming around those chromosomes, and a cell plate forming down the middle. So this stage must be telophase. Press pause if you need to. So that brings us to the end of our cell cycle video for eukaryotic cells. Make sure you have these vocabulary words: cell cycle, chromosome, chromatid, cytokinesis, homologous pair, interphase, and lastly, mitosis. Don't forget your 3 R's. Review your notes. Make sure your drawings are good and detailed and you've labeled everything. Reflect on each page of your notes by doing a 30-word summary, an illustration, or a graphic organizer on the opposite page. And finally, respond to this question. Compare the cell cycle of prokaryotes and eukaryotes. How are they similar? How are they different? Use the words prokaryote, eukaryote, binary fission, mitosis, interphase, and cytokinesis in your answer. You could put your answer on the last page of your notes or as a reflection on the opposite page of the last page of your notes. Thanks for watching.